So today's lecture is on Chapter 17, titled From Gene to Protein. And certainly will be very helpful to view the PowerPoint presentation and, of course, to read the chapter on this material to have today's presentation be in a more comprehensive context. Again, we're reminded that we're performing highlight features here, if you will, on these topics for the chapters. And it's probably even more relevant for this one today. I'm just trying to draw a double helix, DNA double helix here, just quickly to remind us that somewhere along the double-stranded DNA of a single chromosome of a homologous pair, we have an allele, and now we're just talking about it as a gene. And we recall that repeatedly we've stated that from a gene you get protein. We back off from that now to remind ourselves that from a gene we get RNA, and there's a wide array of different types of RNA molecules, and they have various diverse functions, one of which is performing like an enzyme as a protein does. So we'll see that today. But we're focusing right now on getting from the gene to the gene product, the protein. So there's a process, as we know, of getting RNA. I'm not putting messenger RNA here, but it's the one that we're thinking about because there's differences in how the mature mRNA is produced between prokaryotes and eukaryotes. So in essence, we are thinking of messenger RNA, and the process to get from gene DNA, obviously, to this initial transcript, as it's called, is called transcription. So you have a code, you hire somebody that can transcribe that into an intermediate message, and then that message can be used to generate something that the general population can understand, if you will, and you hire a translator for that part. So the process from RNA to protein is translation. Now there's, in eukaryotes, uniquely, there's RNA processing as part of this overall transcription to get to the mature messenger RNA. In prokaryotes, you'll see it's immediately available as messenger RNA. Translation is, in essence, the same in prokaryotes and eukaryotes, but then after the protein is initially produced, it will undergo processing. We've talked about the different structures of proteins before. And we start first by reminding ourselves that one prime example of a protein or a gene product is an enzyme. And this is what got the whole story started, and that's where we're going to step on to next, is to ask the question, find out where I am here, zoom out just a bit. How did we come to know? So you've already discovered in this course throughout the semester that we are learning history in the process of learning about the biological concepts. So as we study who did what and what they discovered, we come to know and understand, hopefully better and more readily, the concepts. And the concept we're talking about now is getting from gene to protein. So how did we come to know this? Well, it started, of course, all along the way, but one of the milestones was around 1909 or thereafter, so early in the 20th century, is that right? There was a physician, Archibald Garrod, who had observed in patients, I see many patients, and in looking at various problems, especially where there'd be particular metabolites in the urine that shouldn't be there, that aren't there in otherwise normal patients, and asking questions about how could the body produce this molecule rather than some other molecule. And so in asking these types of questions in his mind, and of course with studies, of course, is similar to this one, but not quite as detailed because they didn't know as much. But he postulated that the symptoms of some inherited diseases reflect an inability to make a particular enzyme. So at this time, enzymes were relatively well understood. We knew that it took a substrate and changed it to make a product or products, and that there might be series of enzymes to produce things. So the idea of an enzyme metabolizing molecules, changing one thing to another, was already present in their minds from their studies of biology as it existed at the time. That a G, G-A-R-R-O-D. So Gerard then hypothesized that inborn errors of metabolism, as they were called, for example, e.g., alkaptonuria, so as the name implies, there's something showing up in the urine, an alkaptone, that isn't normally there. So how did it get there? And they knew that it occurred in families. You know, the understanding of pedigrees was quite good at the time, so it could be recognized that it was an inborn error being inherited. Oops, OCC occurs because the genes dictate the production of the specific enzymes and affected individuals with these inborn errors of metabolism have genetic defects of course that cause them to lack certain enzymes. Now it's actually to lack functional enzymes, or enzymes that accomplish the appropriate task, if you want to think of it that way. There may be some genetic mutations that cause genes not to be expressed at all, but everything that we've looked at so far is that the gene will be expressed, that particular allele, and the gene product will be there, and if the mutation is such that it changes the protein, then the protein might not be functional, or it might function in a completely different way, although still with the same similar endpoint as we've seen with putting carbohydrates onto proteins that are on the surface of red blood cells. But we could state it the way that we just did. In essence, clinically, they lack that certain enzyme. Well, in the 1930s, a couple decades later, Edward Tatum and colleagues So with a couple different groups, he studied a couple different things. First, they used Drosophila, the fruit fly, and what they did is they associated mutations in the eye color, as we had talked about, with faulty enzymes that are responsible for pigment synthesis. So this was critical because the association was made with the expression of the gene, getting the gene product, the protein, but it just doesn't do what it would be normally expected to do. Also, as his studies progressed and he started working with other colleagues, they used bread mold Neurospora C-R-A-S-S-A, Neurospora crassa, genus and species for that. And what they did is they utilized X-rays, inducing X-ray mutations in the molds. So there are mutations of genes for biosynthetic enzymes. And this is an interesting story, and it's covered in detail in the chapter and in the PowerPoint presentation. And so ultimately, ultimately, came up with one gene, one enzyme hypothesis. Well, of course, now we have to adapt that. Not all gene products are enzymes, and not all proteins are big proteins, so they use the term one gene, one polypeptide. And the textbook uses the term hypothesis for that, but now it's so well substantiated, we might even consider it certainly a theory, but even a law. We won't debate that to any degree. So there is a special notation here, but one gene can lead to more than one isoform in eukaryotes. So there are some advances on that idea. Remember that when we use the term gene product, we're meaning protein. And the process in eukaryotes by which one gene can produce more than one isoform of the gene product, the protein, is in the processing of the RNA between DNA and the mature messenger RNA, where there's events called alternative splicing, and we'll talk about that. Well, now let's kind of get a bit specific and remind ourselves that the key players are the nitrogenous bases. Right? Without these, there's 4 of them in the DNA. Slight differences, as you know, between the DNA and RNA. But just the same, the nitrogenous bases are the key thing. They are in a particular sequence in the gene to determine the sequence of the DNA within the gene to determine the sequence of nucleotides in the RNA, messenger RNA, and then that sequence of nucleotides in the RNA dictates the sequence of amino acids in the protein. So that's the whole idea that we're working with. Well, let's take an example of a nitrogenous base. What about adenine? So where might we find adenine? Definitely DNA, definitely RNA. Any place else we use the A when we talked about nitrogenous bases? ATP, right? Adenosine triphosphate contains the adenine nitrogenous base. Remember, there's for a nucleotide, there's the carbohydrate, either ribose or deoxyribose for RNA or DNA respectively. There's the nitrogenous base, adenine is one of them, and then there is the phosphate group. Well, what about alpha glucose? Nah. What about in proteins? Nah. What about in carbohydrates? No. What about triglycerides? No. And what about hormones like testosterone? No. The nucleotide bases are very important for encoding proteins at the level of the DNA and at the level of the RNA. And they're also useful for other mechanisms within the cell, like storing energy. Another one that we hear about, it doesn't have adenine in it, it has the guanine in it, GTP. GTP is actually consumed in protein synthesis, and it's just another form of high phosphate bond for the chemical energy. So this reminds us that energy is consumed for the synthesis of proteins. It's one of the major cellular actions where we consume energy. So it's an endergonic reaction, right? Energy goes in to the, or energy is consumed to achieve the work that is done. So then, for the encoding of proteins at DNA and RNA, the nucleotide sequence, is also key. And this is true for both prokaryote and eukaryotes. Right? Even in the microbes, the DNA sequence of nucleic acids dictates the nucleotide sequence in the RNA dictates the amino acid sequence in the proteins. So that basic concept is the same in both prokaryotes and eukaryotes. Where they will differ is how they produce the mature RNA, mRNA, messenger RNA, that can be used to make the protein with translation. And we'll bring about those distinctions, but this is certainly one of the commonalities. So, for example, in DNA, we might find along a gene the nucleotide sequence of ACC on the template strand. The template strand is the one being used to make the messenger RNA. So not both strands are used to make the RNA for any one particular gene. Remember, we have two chromosomes in a homologous pair. So we've got the allele on each chromosome. On one of the strands of the DNA, it's going to be the template strand where we're transcribing to produce the messenger RNA for each of those alleles in that pair. So remember, for each gene, we have two homologous chromosomes. There's one double helix. We have another double helix. This might be chromosome number 5 or something like that. And there's some gene on it. And this is the template strand for the gene on this maternal chromosome. This is the template strand that's used to make the RNA on the paternal one. And so these are the two alleles for that particular gene. So we always need to go back to that and remind ourselves. Textbooks always kind of wander away from that point and then lead you to believe that there's only one gene in our genome for a protein that's being transcribed, but there's two of them, one on each homologous chromosome, maternal and paternal. So we go back to that. So let's redraw that as I kind of went off a bit, but it's always important to reinforce that idea when we think about, when we try to go from gene to dominant and recessive traits and all the stuff we looked at before. So again, let's get to this DNA and think of the importance of the sequence of the nucleotides. And what it turns out to be important is that in triplets, they encode a unit of information. So again, this is the template strand of the DNA. And as the RNA is produced, the enzyme doing this, the RNA polymerase, as we'll see, puts in the RNA type of nucleotide that has ribose sugar, and we'll put a U instead of a T into the sequence, the nucleotide sequence of the RNA, and these three that correspond to those three are going to encode for a particular amino acid, in this case, tryptophan. We've all heard about tryptophan. It's the one that's high concentration in turkey and milk, and so if you have a glass of milk at night, it's supposed to be promoting drowsiness and sleep. Anyway, so this triplet is called a codon. So you notice that I'm wearing my jeans today, but I don't have my codon. It's too hot. Horrible, horrible comedian. But I try. So it is 3 nucleotides. A codon is 3 nucleotides long. And this is really important. Where the DNA site is to begin transcription so that your messenger RNA starts at a particular nucleotide so that the appropriate sets of 3 are used to put the appropriate amino acid in the protein is very important. Changes in those mutations, as we'll talk about at the end, can have devastating outcomes, or maybe no outcome, no obvious effect at all phenotypically. Anytime there's a change in the gene makeup, nucleotide sequence, or the composition, that's a change in genotype, right? But once you start looking at the protein, what it does as an enzyme, say for example, or what it contributes to the overall appearance of the animal, then you're talking about phenotype. So back to this codon. So a codon may code for the same amino acid. From here on out, I'm going to use uppercase A for the abbreviation of amino acid, as another codon. And you ask, of course, how can this be? Well, of all the possible combinations of these triplets to make the codons, it turns out that there are 64 possible, but only 61 of the 64 possible triplets actually code for amino acids. And how many amino acids are there? There are only 20 amino acids. So there's obviously going to be more than one codon for particular amino acids. What this is called is redundancy. So thus, there is redundancy. More than one codon is used for a particular amino acid. So I'm going to label this with an A in a circle for our later comment. A big but here. Whoops. There's no ambiguity. Meaning that even though you can have more than one triplet encoding for an amino acid, there's a built-in mechanism so that it'll never encode for a different amino acid unless you change the nucleic acid sequence, right? Like with a mutation. So there's no ambiguity. Anytime, if for any one amino acid, as we'll see, if any one of those codons show up, it's always going to put that one in there. It's not going to confuse it for another amino acid. Well, it turns out for the tryptophan, TRP, the tryptophan example that we used, there actually is only one codon. And it is UGG, as we saw. So we started with an example that didn't fit the rest of our discussion. But let's consider proline. Can you have a CCU or a CCC? Start noting that these first two are going to be the same in all of these codons that are redundant for this amino acid. CCA and CCG. So the first two will always be unique for a particular amino acid. That is the control for the B in the circle above, the ambiguity. Right? So even though there's more than one codon for an amino acid, there's never any confusion because the first two are always the same. The third one is what changes. So it's that third nucleotide in the triplet codon. It's the third that differs. So that third one differing is what accounts for the redundancy, A in a circle, labeled as redundancy up here, right? So you can change just the third one a couple different ways and still encode for the same amino acid. Now this one also is very important for mutations, when considering mutations. Because if a codon in a gene is originally CCU and there's a mutation and it's replaced with an A, you don't change the amino acid. But if you change any of the first two, then either one of them or both of them, then you will change the amino acid in the protein for that codon. So that's what's important about that level of discussion there. Well, now let's get to a grander scale of things and think about transcription in general and how we get from the gene to the RNA. So I'm going to draw a straight line that represents only the template strand. So as we simplify our models, we've gone from two chromosomes of a homologous pair, each having a gene that is one of the alleles for a trait of a character, to talking about a gene within a double helix, one of the strands being the template strand. Now we're just drawing the template strand. So don't lose the overall complexity as we get to this simplicity. So again, this is just the template strand used to make the RNA. And this is the 5' end, and this is the 3' end. Standard way of drawing the template strand. This here represents the start site. I might use start point in the textbook. I can't remember exactly what word they use, but I don't think they use start site, but that'll work. And to the left of that, to the 3' end, is called upstream, going upstream of the gene itself. The coding portion is here. And then to the right is downstream of the start point. It's even, just quickly, it's actually, the nucleotides are given numbers. So start site is the 0, and then it goes like a number line, positive to the right and negative to the left. So it gets more detailed, as you might expect, but don't get too concerned about all that. So the positive number side here, or downstream of the start site, is the actual gene that we would think about that will result in the protein. And upstream of that is called the promoter. And there are proteins, a variety of them, they're discussed in detail, bind to the DNA or bind to each other and affect transcription. And proteins that bind to DNA and affect transcription are called transcription factors. And these are very important. We'll study them in more detail. You see it in the PowerPoint presentation for regulating the initiation of transcription. And later we'll talk about regulating gene expression, period, whether it's expressed in a cell or how much it is expressed in a cell. In the promoter region, there's another portion that has nucleotide sequence TATA, called the, can't read that, but it's called the TATA box. That's also in the promoter region. It's usually pretty close to the start site, just upstream. And with the appropriate transcription factors in place, in association with the TATA box, the RNA polymerase is going to bind here. So this circle is the RNA transcriptase. So it's a complex of proteins, but one of them within that complex is the actual enzyme that's going to read the nucleic acid sequence in the DNA and generate the complement sequence with the appropriate nucleotides, like U rather than T, in the RNA. So that sets up our basic idea of how transcription is initiated, and it proceeds from the 3' direction to the 5' direction. So it generates a complement that is anti-parallel. So the messenger RNA would be produced starting over here on our left, the 5' end, because it's anti-parallel. If this is 3' for the DNA, then you're going to have 5' end of the RNA strand there. And it's going to be assembled in this direction, 5' to 3'. And it'll initially be the full length of the gene. Some of those other codons are start sequences and stop sequences, so that's what the other 3, remember 61 out of 64, the other 3 work as those and some other types of things. So the RNA is synthesized from 5' to 3'. The And the RNA polymerase binds to the promoter region, right, in association with the Tatabox. To begin transcription. That's called initiation. And we'll have a few more discussions on that. So everything we've said so far is true for both the prokaryote and eukaryote. So far we're true on both of them, and we want to go on to talk about big differences. So we know structurally, that S looks horrible, there's distinctions between prokaryotes and eukaryotes, and that contributes to the differences in how things work for protein synthesis overall between the two types of organisms. So let's consider first the prokaryote to make one point. There will be a few other points that we will make on these big differences. Remember, no nucleus or other membrane-bound organelles, right? So transcription and translation are coupled. Let's draw a little illustration of this first, and then we'll make a couple of other points. But first, we say, therefore, there is no RNA processing as we will see in eukaryotes. So as soon as the RNA is made from the DNA, it's immediately available for expression, for translation. So here's the DNA. Again, the template strand. Somewhere there's a promoter region, transcription factors, gets things started. Here's our circle showing the RNA polymerase. This is the template strand of the DNA. Several RNA polymerases, as in eukaryotes as well, can proceed along the gene as they're, of course, proceeding in the same direction, 5' to 3' along the DNA. No, said that backwards. 3' to 5' along the DNA, and therefore making the RNA first at the 5' and then toward the 3'. So as it's proceeding, it's making longer and longer messenger RNA. And so as soon as it's available, a ribosome can jump on it and start making a protein. So this one might have a second ribosome a little bit farther along, and then the polypeptide is going to be longer. This one might have one that just got started, one that was farther along, one up here. And so you've got, these are all supposed to be, I keep getting longer and longer. Okay, the one closest to the DNA has been on the messenger RNA for the longest time, so it should be the longest. So let's put one way up here now on this one, and there's the, and of course this is the polypeptide growing coming off the ribosome. And these little black things are the ribosomes. Right? So with the prokaryotes, as soon as the, and of course we'd have all the others, this messenger RNA has been there the longest, so we're going to have more ribosomes on it. So this is the first part of the messenger RNA that was produced, the 5' end, way over here when it first got started. So here's that 5' end on the newest piece of messenger RNA. And as soon as the site for the ribosome to bind to it is completed, ribosomes are there and they'll jump on it. Amino acids are available with everything else that we'll talk about, and the translation begins immediately. So what this means is that what comes off the DNA is the real mRNA. In eukaryotes, it isn't. It has to be processed. So in prokaryotes, the mRNA transcript these are different lengths of the same mRNA transcript being transcribed. The mRNA transcript is an exact copy, an exact complement of the gene from which it was copied. Right? In eukaryotes, that first transcript that's produced, called the initial transcript, is going to be processed, and we'll look at that in a moment. And then within the nucleus, then the messenger RNA ends up in the cytoplasm. So if you had the sequence for messenger RNA and tried to use that sequence to find a specific sequence in the DNA, you wouldn't be able to find it because it isn't exact for the particular linear sequence. So it sounds a little confusing. Let's then illustrate a eukaryotic gene. And again, we are drawing the DNA of the template strand from 3' to 5', promoter region to the upstream of the start site. And then we put in these boxes. I'm going to put in 3 of them with some intervening sequences. These are called exons, exon 1. And the intervening sequences of DNA, the box is just illustrating a length of sequence or length of a sequential nucleotides, and it's not thicker in that area in the gene or anything. It's just illustrative purposes. And this is intron 1. This is exon 2, intron 2, exon 3. Right, so there's going to be a start site here for transcription. There's also a sequence that gets turned into the RNA that becomes the start site for translation. And then there's stop sites over here for stopping transcription, and one that also gets there in the RNA to stop translation. So when you read the text, they'll talk about transcription start and stop sites and translation start and stop sites that you can find on the DNA and thus on the RNA, and they have their particular functions. So the initial transcript from this, once the appropriate transcription factors are present and the RNA polymerase binds, we get our initial transcript. It's going to be the exact length from start site to the stop site. It'll be 5' to 3'. And I want to use a couple of different illustrations. Again, these are just illustrative purposes to say that along this linear sequence right here of nucleotides in the RNA, we have the complement of the sequence of nucleotides within the exon 1 DNA. So I'm just going to put the 1 there, and then I'm going to slant to the right for exon 2 being represented on the RNA, and then slant to the left for exon 3. Oops, just putting the 3. Okay. So they're no longer called exons, but the complement of the sequence is there. So this is called the initial transcript, or primary transcript. So the sequence of the primary transcript, or initial transcript, is the exact complement of the eukaryotic gene, but it is not functional messenger RNA. There has to be RNA processing. So this initial transcript is not ready for translation yet. And this is eukaryotes, remember. It must be processed. So we ask ourselves, what are the processes? Well, let's fit in a definition here first in words that you might find useful for exams, of course. Let's spell it correctly. Exons, it's not where you get your gasoline. Exons are the coding sequence, the coding segments, or the coding segments, so it's just a segment of the entire gene, right? So it's segmented in that sense. The coding segments of a stretch of eukaryotic DNA. So exons are the coding segment of a stretch of eukaryotic DNA, just as we had described. So from here we'll go on to talk about transcription. We'll discuss transcription, and then we'll talk about the processing of the RNA primary or initial transcript. So with transcription, as with translation too, initiation, elongation, and termination. There are 3 stages of it, right? Put this on there. Termination, okay? So initiation, elongation, termination. So at each point in discussion, I'll try to refer us back to which stage we're talking about. Let's start first with, of course, initiation. And again, the details are in the PowerPoint presentation. So I'm skimming over this, assuming that you either have seen that or you will go and view it afterward and maybe come back to this if you need to have further clarification of these points. Eukaryotic transcription initiation complex. So it does differ from prokaryote, but there's a lot of things that it shares. And let's just now focus on eukaryotic. There's a promoter. There's a Tata box, remember that. Got to be transcription factors. Got to be RNA polymerase. There's different kinds of RNA polymerases in eukaryotes because they make a bunch of different kinds of RNA. What about lowercase n, s, lowercase n, RNP? These are small nuclear ribonuclear proteins. Ribonuclear proteins. And that gives us the SNRNPs. Okay? Well, these are not part of it. I'm just bringing it at this time. Well, hint, hint for the exam question. Which of the following is not true about the eukaryotic transcription initiation complex? If you don't see that question, exam question built into that series of transcription on my part, then you're missing our whole point about these recorded lectures. This is functioning differently, and let's kind of make a transition to get a feel for that. What is a ribozyme? Ribozyme. So it hasn't been very long, decade or more, I guess, that we've come to understand that nucleic acids, actually RNAs specifically, have enzyme-like activities. They can act on a substrate, have a specific binding site. They can change the substrate so you get a product. So it's RNA in association with proteins, therefore riboproteins, ribonuclear proteins. But it's the RNA within the protein RNA complex that's actually acting like the enzyme, binding site, making the changes in the substrate to get a product. So it is an enzyme made up of RNA. But as I said, it's complex. It's in a complex with proteins. So let's think about these SNRPs a little bit more. So the SNRNPs. They are involved in the RNA processing, and RNA processing occurs only in eukaryotes, right? So these little SNRPs are made up of both protein and RNA, as we said. They join together to form a large structure called the spliceosome. So multiple SNRPs get together at the junction of introns and exons to form this structure that's going to bring about the splicing out of the intron and reconnecting of the exons, as we'll see. So they join together to form a complex. Oops, to form a large structure. Sorry about that. It is complex, but let's keep our words so that they're recognizable. At times you need to recognize these sequence of words. So it's a large structure called the spliceosome. And to bring about the splicing of one of the processes, obviously, is splicing of the RNA primary transcript, and the SNRPs will bind to the splice sites at each end of the intron. So just quickly, we had the template strand with the exons and the introns produce the message. I'll use another designation for the exons here. So this is the intron. This is exon 1, exon 2. So this is intron 1. So this represents it here. So at the junction of the intron and exon in the transcript, this is where you get the binding of the SNRPs, and then other ones are going to come in and bind, and eventually it'll form a little loop there, bringing the exons, my loop twisted, but it's floating around in solution within the nucleus. So you get this complex there so that the intron can be excised, cut out, and then you splice together the exons at their two ends, where they used to connect to the intron in between them. So the illustrations from the textbook are very useful for that. If you want to get clarification, go to the textbook or a PowerPoint presentation. Let's now consider RNA processing in total. And again, eukaryotes only. Oops. Started to write only within eukaryotes. Eukaryotes only. Make that look like an L to correct my mistake. So there really are three examples or three processes of RNA processing. There's a 5' cap that's added as the transcript becomes available. Remember, the first part of the message of the RNA, to be produced is the 5' end. So a cap is added there. As soon as it's done, then a 3' poly A tail is polyadenylene tail is added. So these are added after transcription. And so because I'm just using the word transcription, we're talking about initiation, elongation, and termination. So shortly after initiation, the 5' cap can be added, but only at termination will the 3' end be available, so then the poly A tail is added after the termination of transcription. So we just used the term transcription there. And the third example is that splicing that we just described. So with splicing, the intron segments represented in the RNA, the intron in parentheses RNA nucleotides are removed. And the exon portion of the RNA, called the codon sequences, are spliced together. So for splicing, you have to have, there are two parts of splicing. You have to have an excision, cutting, right? If you're going to have a piece of rope and you need to take a piece of it out along its length, you got to take some scissors and cut it at 2 points where intron joins exon. Then you have to ligate or get it stuck back together, glue it or tie the two rope ends together after you've cut out the segment, right? So these two parts of it, you have to get the excision and the ligation. So an enzyme ligase is going to come in and ligate it, but we ask what causes the excision, and we'll talk about that in just a moment. Why don't I write it right now? So during splicing, RNA is a molecular component so the RNA is the molecular component of the spliceosome that catalyzes the excision reaction. So RNA acts as a ribozyme to bring about this cut in the primary transcript. Well, what about what is the significance of the 5' cap and the poly A tail? So what do they do? Well, helps to stabilize the mRNA. By inhibiting the degradation. Right. So this is pretty common for the production of molecules. You make the first form of it so that it has something in it to protect it, to keep it available long enough to do its job, and then ultimately that protection wears off, the molecule wears out, you degrade it, and you recycle the materials, and you bring in fresh, new molecules, part of the life cycle of molecules. So the 5' cap and the poly A tail contribute to that. It's also thought that they contribute something else. So let's draw our now mRNA. If we could go back to our initial transcript before, we would see that this one is shorter because we have removed the intron. So we've got the 5' end of our RNA now after processing. 5' cap at the 3' end, a poly A tail was added to it. And if we looked at the nucleotide sequence, we'd have exon 1, back to that original one where we used these illustrative marks to show it. Uh-oh. I'm going to have to extend them just to, might not seem the same length, but these are illustrative purposes, remember that. So we would see the complementary sequence to exon 1, exon 2, and exon 3, even though I keep changing the way I write my E, those are all E's for exons. But no introns would be present. So this, after RNA processing, the 3 stages, this is eukaryotic messenger RNA. It's not the exact sequence of the gene from which it came, but in the prokaryotes, we saw that the messenger RNA is made immediately without any processing. So the production of mRNA that now is got permission to get out of the nucleus and ready. It is into the cytoplasm. It is ready to meet up with a ribosome and for translation. The prokaryotes, when they make it, it's already basically in the cytoplasm, and it's immediately available for ribosomes for the translation to occur. Well, before talking about translation, let's think about a mechanism by which there is greater complexity and diversity of proteins than the number of genes that eukaryotes have. Therefore, it's thought that that's part of the significance of having the introns and the RNA processing. And that is called alternative splicing. So we followed it through where exon 1, exon 2, and exon 3 remained represented in the messenger RNA. The same gene might, under certain conditions, be spliced in an alternative way so that only exon 1 and exon 3 ended up represented in the messenger RNA, and you're going to have a very different protein. Or maybe just exon 1 and exon 2. And of course, some genes for proteins have many exons, 5, 20. I don't really know the highest number, so I'm just going to shut up on that one. But it's more than 3. So alternative, I forgot to put the RNA in there. RNA splicing can allow the production of proteins, different isoforms of proteins, of dramatically different amino acid sequences. There's AA for amino acids. From a single mRNA. Now this is the wording that I want you to, sorry, I had it off screen for a minute. The use of mRNA there is what I want you to recognize, but I also want you to recognize that that is wrong. It's different amino acid sequences from a single primary transcript that can become different forms of messenger RNA. So I've already told you that I'm borrowing these questions from whoever wrote them for the textbook company. And here's one point that I disagree with the term that they put in the question, but recognize it for what they're trying to mean in general. But we just discussed the specifics, and we know that they really should have used RNA transcript there rather than mRNA. But we'll give them that at this time. So it can allow the production of proteins dramatically different, kind of the same idea as the first one, from a single, at least they were consistent, mRNA. So again, we don't appreciate the utilization of that term here because it just isn't correct. So let's just make a little notation, or underlining there, asterisk or whatever. The use of the term in these statements of mRNA is referring to primary transcript. So we kind of take things literally to be as accurate as we can be. This one time we can apply a slight variation on what we're understanding when we see mRNA to appreciate the alternative splicing. But you can see that if a portion of an exon or a whole exon is not included, you've got a totally different amino acid sequence from the same gene in the gene product, and dramatically different protein. Now let's work on translation. So the following are directly involved in translation. So this is an important word. When you see directly, you think of the closest relationship between A and B, if you will, as possible. Well, obviously, messenger RNA is. Transfer RNA is bringing the amino acids in to the ribosome. The amino acyl tRNA synthetase enzymes are responsible for putting the appropriate amino acid onto a specific transfer RNA. And then, of course, we know we need ribosomes. Well, what about DNA? Obviously not. It's very indirect. It's far away from, well, if we're thinking both prokaryote and eukaryote, it's not necessarily far away distance, but it's remote from that process. The DNA will make the RNA, and then it's no longer involved in making the protein. It just gets out of the business at that point. So what is this transfer RNA? What does it look like? How is it made? What are ribosomes made of? Let's just highlight a couple of things here. So transfer RNA, tRNA, looks like this. It's transcribed from a gene somewhere by a different RNA polymerase. And then it forms this kind of a shape, two-dimensionally. Three-dimensionally, it curves even more to look like an L. But at this end here is where the amino acid will be bound to it by the amino acyl tRNA synthetase enzyme. And there's one of those enzymes for every amino acid. And then at this end, in this loop down here, there's 3 nucleotides in a specific sequence. It's loop 1, loop 2, and loop 3. So in loop 2, there's these 3 nucleotides, and they are called the anticodon. So they are, the anticodon is part of transfer RNA. And of course, it's the complementary sequence to a codon within the messenger RNA. So it recognizes the codon in the messenger RNA and brings the appropriate amino acid to the ribosome to be inserted into the protein at that point. Well, here, I wish I could draw squiggly lines. Hopefully you can see those as squiggly and not straight. I'm representing hydrogen bonds within the molecule itself. So the hydrogen bonding in those locations is between base pairs within the molecule, and they are responsible for maintaining the shape of tRNA molecule. Well, let's think of one other type of RNA first and make a couple points about that real quick. What about ribosomal RNA, or lowercase r RNA, ribosomal RNA? So ribosomes are also riboprotein complexes, but they're found in the cytoplasm of eukaryotes. Well, in eukaryotes, remember, they're transcribed in the nucleolus, which is in the nucleus, right? Nucleolus, and that is in eukaryotes, only eukaryotes, because only eukaryotes have the nucleus with the nucleolus, right? And it's the most abundant type of RNA. Why is that? Well, because there's more than thousands, or many thousands, of ribosomes per cell. And each ribosome has got a few pieces of ribosomal RNA contained within it. Well, let's get the ribosomal RNA, messenger RNA, and protein and transfer RNA linked together. We'll build on that idea. Let's first zoom in on where a ribosome would have that loop 2 of the transfer RNA in it. So this here is the ribosome. We've zoomed in. Ribosome. And this is that loop 2 of the tRNA. And we're looking at the anticodon on it. And then also within the ribosome, the messenger RNA would be sitting there, and there's 3 nucleotides on that. So on the mRNA is the codon. On the transfer RNA here is the anticodon. Transfer RNA is going to bring a particular amino acid that's specific for this anticodon, and when it can fit into the piece of messenger RNA along this part of the ribosome messenger RNA complex, then it's going to bind to the ribosome because it matches the codon in the messenger RNA and bring in the appropriate amino acid. Now here, because we're already partly down the messenger RNA, this would be the elongation part, right? Way over here would be initiation. We're here at elongation, somewhere above a protein, a polypeptide is being produced. And then the stop site's going to be, the translation stop site's going to be over here someplace, and the ribosome gets there and it's going to drop off. Well, let's look at the ribosome at a lower mag right now. It has 2 subunits. Has a large subunit with 3 sites on it. These sites are the A site, the P site, and the E site. So A, P, and E sites in the large subunit. In between, and then this is the small subunit here. And the messenger RNA would be lying there in between those. So up here, what we didn't draw in is there should be the small subunit of the ribosome somewhere here holding the messenger RNA for the tRNA to come in. So this is the mRNA, and translation is going along that way. We're somewhere along the length of it, so this would be during elongation. And the transfer RNAs would be coming in, delivering the amino acids, getting the amino acid linked to the growing polypeptide chain, and then the amino acid, the tRNA, excuse me, that no longer has its amino acid because it was just added to the polypeptide chain, has to exit. And it'll jump along from A to P to E. Walks along with its knuckles dragging on the floor, if you will, like an ape. Transfer RNA. So, as a ribosome translocates along an mRNA molecule, as a ribosome translocates along an mRNA molecule, by one codon, So we drew it associated with a single codon. What happens is the tRNA that was in the A site moves to the P site. Okay, well, since we were looking at it in the elongation phase, and you look in the text in the PowerPoint, talked about initiation and termination of translation. We're thinking of elongation, and it's you really need to look at those illustrations to get a better, more complete feel for it. We're just taking little excerpts here to highlight. So for the tRNA that was in the A site to get into the P site, the tRNA that was in the P site moves to the E site, so APE, right? And from the E site, it is released. So we said we got a new one's got to stick to the A site. The one that got there just before it moves to the, or is in the P site, holding the growing polypeptide strand. And the one that was just before that one that's now in the P site is in the E site and being released. So it's APE. So now let's try to think of a sequence of events of protein synthesis. So a small ribosomal subunit binds with the mRNA. So I'd stated that the small subunit is the one that has the site for the mRNA to bind to it and holds it in place. So that happens first. So now we're beginning to think about initiation, right? And we put in a box here to remind ourselves that after that, the large and small subunits unite. In quotations. Right? So the small and large got to come together with the mRNA in between them, and then the APE sites are in place. So next, an amino acyl tRNA. That means a tRNA loaded with an amino acid by the enzyme synthetase that we talked about, right? Binds to site A. A peptide bond is going to be formed between its amino acid WEEN. So this new amino acyl tRNA that just came in, we'll call it the new one. So between the new amino acid and a polypeptide chain, so the polypeptide chain is now stuck to the amino to the tRNA that got there just before the new one we're thinking about, got shifted to the P site. It has the peptide or polypeptide stuck to it. You can link that with the P. And then the one that was in the P site before hit the E site and has been released. So this ratcheting thing is kind of hard to put in total words, but bring a new one to the A site, and then the amino acid on it forms a peptide bond. It's added to the polypeptide chain. this tRNA now has the polypeptide attached to it, and so it's got to shift from the A to the P site. So that tRNA, not calling it aminoacyl anymore because it doesn't really belong to the amino acid. It's still stuck to it, but it doesn't belong to it because the amino acid has now had the peptide bond formed. So the amino acid is really part of the polypeptide or growing polypeptide chain. So that tRNA, the new one that we were just talking about, will translocate or translocates to the P site because it now has the polypeptide attached to it. Then, as this process continues, this tRNA will end up giving up its connection to the polypeptide as the next new aminoacyl tRNA comes along to begin this process at the top. We're following just this one tRNA coming through. So it had the polypeptide attached to it while it was in the P site. It's going to give it up to the new one coming into the A site, and therefore it's going to leave the P site as it gives up the growing polypeptide, and momentarily at least, the P site remains vacant. So there's just a little skip in the transition where that P site's going to be available for just a moment. Whoever knows how long it might be for the process that we talked about here to continue on. But as soon as this one that just came in is ready to translocate to the P site, the one that was there before has already gone to the E site and is being released, and the P site is already vacant for it. So there's just a moment there where the P site will be vacant. So in eukaryotes, the translation is in the cytoplasm. After transcription and RNA processing. And of course that completed in the nucleus. That's one thing that the nuclear pores do. They allow things to move in and out. The mRNA has to move out after processing. So there is extensive RNA processing that's required in the eukaryotes before the transcript. That should be a B. Can be translated. Also, we could say that eukaryotes have complicated mechanisms for targeting proteins to the appropriate cellular organelles, subcellular organelles. Remember, the eukaryotes have the membrane-bound organelles, ER, vesicles, Golgi, and so on and so forth. And there's particular mechanisms for getting those proteins there. There has to be, because those things exist. Those organelles exist in eukaryotes, and they have proteins inside them or within the membrane And of course that's discussed in greater detail, a particular mechanism for that, to get it to the ER, in the textbook, in this chapter and the PowerPoint presentation. We had talked about it in general before when we studied the cell and the organelles. Again, protein synthesis in prokaryotes the translation, so contrasting now to what we just said in eukaryotes, translation can begin while transcription is still in process. Still in progress, excuse me. So you can go back to the illustration we drew to reinforce that idea. Then finally, just a quick consideration of DNA mutations. Now that we understand codons and redundancy and lack of ambiguity, with DNA mutations, there can be point mutations. Well, first, let's say that there can be deletions and substitutions. Right? And these can be one or more nucleotides. Right? If it's only one nucleotide, then those are point mutations. And we saw how those are actually most likely to occur on the third nucleotide of a codon, and may not even, because of redundancy, may not even change the protein ultimately. But what if you had a codon, a full set of 3 where you had a deletion, or a codon substitution? So 3 in a row that happen to be the codon. Well, what would happen here is it would only be one. It only affect one amino acid, right? Because it's one codon, and it would be either a loss with the deletion or substitution for a codon substitution. So we know that with sickle cell disorder, it can have a big impact due to pleiotropy. But in general, for the protein, if you just looked at the protein, it's just one amino acid that's changed. So the chances that it's going to have a big effect on the protein and ultimately the phenotype of the organism are less than if you have what can cause a frame shift type of an effect for reading the nucleic acid sequence. So a good example of this would be a base pair. 2 separate words, base pair deletion. So a base pair deletion, gotta go to another page. A base pair deletion would be most likely, or is most likely to be damaging to the protein it specifies. So we know along the DNA, 1, 2, 3 for codon. I'm going to put a little space in there, but realize that they're right next to each other. But just so we can see them in triplets, I'm separating them. You know that in the DNA they're exactly side by side. So here's our codons. You take out one of these guys, and then from that point on, everything shifts over to here. Right? So there's a frame shift that occurs. So once you change that codon setting, you know, the the triplets that are locked in. These are supposed to be together. These are supposed to be together. These are supposed to be together. So if we had a single substitution there, base pair deletion. I guess you have to check on the base pair deletion. Is it two of them? Causes a significant frame shift so that these three that are supposed to be together and now are no longer going to be together. These two have got to go with that one, and then everything thereafter is going to be read differently. So from this point on, you're going to get a completely different amino acid sequence, right, than the original protein. So it can be most damaging, or is most likely to damage the protein that is specified by that gene. So that finishes our rather long discussion on Chapter 17. I appreciate your time and attention, and we'll see you again soon.